Thank you for joining us. I'm just going to kick off with a karakia, and this is about seeking knowledge. So, e te hui, whaia te mataronga kia marama, kia whai take nai, mahi katoa, tu maia, tu kaha, arahau atu, araha mai, tātou I a tātou katoa. And this means, for this gathering, seek knowledge for understanding, have purpose in all that you do, stand tall, be strong, let us show respect for each other, which is very fitting for this type of speaker series. So a very warm welcome to you all, if you're here in person or online, and this is the third in our 70th anniversary year speaker series, and we are very privileged to have Dr. Tim Angeli-Gordon with us tonight, who will be presenting on gastrointestinal disorders and diagnosis. Thank you for coming along tonight, and as mentioned, we have the 70th year celebrations this year, and we are celebrating over $100 million worth of funding towards medical research from Auckland Medical Research Foundation. And for those of you that are donors in the audience, you were the ones that helped make this possible. So a huge thank you from us. A huge thank you from our research community. You have made possible life-changing differences, so big thank you to you. I'd also like to thank our partners that have joined us for our 70th year celebrations, which are the University of Auckland, and we've got Dr. Frank Bloomfield here tonight, and Nina Bowen, also from the University of Auckland. So thank you very much for being here tonight and for your partnership. Auckland University of Technology are also a partner, along with JMI Wealth. And we couldn't be doing what we're doing this year for the celebrations without them also. So thank you very much. So for those of you that don't know me, my name is Sue Brewster, and I'm the Executive Director of Auckland Medical Research Foundation. Lots of people have heard me say that there's 5 of us in the team and we're all executive directors, because that's how good the rest of the team is. And they're all here tonight, and they make the wheels go round on this wagon, so thank you, team, as well. So, I know why you're all here, and I am not going to take up any more of your time. Now I'd like to invite Associate Professor Vanessa Selak up to introduce our next guest, who is Dr. Tim Angeli-Gordon. Thank you.
Tēnā koutou, ko Vanessa Selak toku ingoa. He aho nuku aho, waipapa taumataro. As Sue said, I'm Vanessa Selak, and I'm delighted to be here this evening at this wonderful lecture series after having heard from Associate Professor Amy Chan, and to now be hearing from another wonderful colleague, Dr. Tim Angeli-Gordon. I am delighted in particular to be introducing Tim, because I've had the privilege of working with him for a number of years alongside working with Tim together on the AMRF Medical Assessing Committee. Currently, Tim is working as Senior Rangahau and Innovation Manager at Te Wānanga o Aotearoa, where he's using his considerable expertise in research to train up our next generation of researchers. And it's incredible to see the amazing work that he's doing supporting that development across the Te Wānanga o Aotearoa. And he's gone from doing research that's focused on the body to research now that's looking at an incredibly broad scape, from down to the cellular level across, right across through to environmental health. However, in terms of his own research journey, that began in the University of Michigan, and that's where he undertook training in biomedical engineering. After doing research there and completing his Master of Sciences, he came to Aotearoa, and that's when he began to undertake research in gastrointestinal health, when he joined the Auckland Bioengineering Institute, which is here with us at the University of Auckland, and that was when he started to do his PhD, and that was in 2009. At the University of Auckland, his research program has focused on developing new devices and techniques that can be used to diagnose and treat gastrointestinal disorders. So I guess we've got a bit of a recurring theme this evening of the use of digital technology, except we're moving from the respiratory system to the gastrointestinal system. So Thankfully, what Tim has been able to do is, in addition to undertaking his senior role at Te Wānanga, is to continue his research, supported by many funders, including the Auckland Medical Research Foundation. So we're incredibly privileged to have been able to have the support to enable Tim and his team to undertake their groundbreaking, world-leading research in gastrointestinal diseases, which he'll be telling you more about. And look, before I hand over to him, I just wanted to say how wonderful it is to have Tim here. He's an incredibly humble person, but don't be fooled. He's incredibly intelligent and wise. And, you know, alongside this commitment is an incredible strength and drive to support Mātauranga Māori and Te Reo Māori and our next generation of researchers. So please join me in welcoming Tim to give his talk. Thank you.
Oh, well, thank you, Vanessa. It's lovely working with Vanessa, and I'm very honored by that introduction. It's always nice and sometimes awkward to be introduced like that. But I feel very fortunate to be with all of you here tonight. Tēnā koutou katoa, no mai haere mai ki te kōpapa nei. Welcome, everybody. Thank you for sticking it out. We've heard about asthma. Now we're going to move, I guess, down the body into the gastrointestinal tract, slightly adjacent, depending on where we're talking about. But typically I make a joke about the gastrointestinal tract or research being taboo, but we've already covered sweaty socks and sweaty shirts, so I'm actually feeling pretty good about my field of research at the moment. So tonight I'm going to talk to you, like Vanessa introduced, about my research in the gastrointestinal tract, particularly coming from my background as a biomedical engineer. Looking at developing new technology and devices from an engineering perspective and trying to translate those through to clinical bedside. And we'll talk about a couple examples at various stages of that process. And yeah, like Vanessa said as well, I've been very fortunate to have a relationship with the Auckland Medical Research Foundation for a number of years now, being supported very early from a fellowship and working for many years on the Medical Assessment Committee. So it's a real honor to be able to be here tonight and to speak with you all. So first, ko waio, who am I? No tia mana ahau, I'm from Germany. Engari I whānau mai ahau me tupuaki ahau ki America, so I was born and raised in America. Ke Waitakere ahau e noho ana I naenei. I live in Waitakere in West Auckland now, in Henderson. Ko Bella Pullum, rawa ko manawa ora, Angela Gordon, aku tamahine. I have two daughters. Their names are Bella and Manawa ora. Ko Joni Angela Gordon, taku hoa rangatira, taku wahine. My wife is Joni Angela Gordon, who you can see there. And I'm very fortunate to get to actually work with her now in my current role. She is also a researcher. She looks at Māori mindfulness, Māori education, and child well-being. And we're doing some quite exciting work now, bringing together my background in biomedical engineering and Western medicine with aspects of Mātauranga Māori to look at gut health from different points of view. So it's quite fun, and I think Vanessa touched on it, but this is a lot of my why. So I'd like to introduce that. And particularly, I think her, which is why none of them are here tonight, because Manawa ora, a 2.5-year-old toddler, it's about witching hour right now. I can guarantee she's running laps around the lounge now with various levels of clothing on. So I don't think you guys needed to see that tonight. And then I guess moving on to my work and who I am in my work. And Vanessa, again, thank you to her for introducing this. So for many years, I did my PhD at the Auckland Bioengineering Institute here at the University of Auckland, and then worked here for a decade building a research program in gastrointestinal health research. And part of that was founding and leading the Target Lab, so a laboratory for translational research in gastroenterology and emerging technologies, which I still lead and run today. But more recently, about 1.5 to 2 years ago, I moved over to Te Wānanga Aotearoa in the Te Manawa Haukura Research Center, which is where I work now. I'm still very fortunate. I work and collaborate closely with the university and many of those partnerships. So I guess it's a little bit of making my own path and figuring out how I can best serve a number of different communities and people. And I think that brings me to the motivation for a lot of this work. There's a very well-known whakatoki Māori or proverb that says, he aha te mea nui o te ao, he tangata, he tangata, he tangata. So what is the most important thing in the world? It's the people, it's the people, it's the people. And that's why I think for me, I like to start my talk about who I am, and really that's more about my fauna and who I'm surrounded by, who I get to work with. But in this case, this is somebody who's very special. So this is Ruby Hill. She is somebody who suffered very severely from a disorder called gastroparesis. So we're going to move more into some of the gastrointestinal health now. And gastroparesis, it literally translates to stomach paralysis. It's a very severe digestive disorder where patients suffer from abdominal pain, bloating, early satiety, which means when they eat a meal, they feel full very early, abnormally early, vomiting, and nausea. So very severe symptoms. And also in this disorder, gastroparesis, the stomach fails to empty properly. So it doesn't empty food like it should. Now, I think many people say a picture is worth 1,000 words, and I also think a face or putting an actual person to many of these disorders can be really powerful. So this is Ruby, and we'll fast forward to, this is also Ruby, not too long after. This is her in the hospital. She was diagnosed with gastroparesis after a very long and painful process with many, many ups and downs. And in the caption there, so these are all photos from online, they're publicly available, but in the caption it says Ruby at her lowest weight of 42 kgs, and that was at the time. So I think very powerful there. And then moving on, you can see, I think probably a more powerful image, and I'll let those images speak for themselves, I think. Unfortunately, Ruby passed away a number of years ago, but I think this is particularly powerful because we actually have Ruby's mom in the audience tonight, Jo Hill, who's here with us tonight. So Jo and I met a number of years ago, and we've been working closely together in terms of just a shared enthusiasm for raising awareness for gastroparesis and working on new diagnosis and treatment options. And so I think to highlight here, this was a very difficult journey. I think you can see that in the photos. And it really highlights the challenges of the diagnosis of this disorder and also the fact that there are limited therapies available. And I think not only is that challenging for Ruby, for patients like her, but it's also challenging for clinicians. I work with a lot of medical doctors and clinicians, and those people really want to be able to treat their patient. They want to be able to give a definitive diagnosis the moment somebody walks in the door. They want to be able to give a quality treatment to make those people better. And oftentimes, the tools don't exist for them to be able to do that in a quick and efficient manner, at least not as quick and efficient as they would like, as the patient would like. So I think in many ways, we're all on the same team here. Everybody wants to deliver good for these patients, and one of the challenges is a limit of options. Oftentimes, that can be technically limiting. We saw from Amy's talk, there's a lot of technical innovations that are advancing asthma, and from my perspective, it's similar in the gastrointestinal space. Now looking a little bit beyond just Ruby and that individual patient level, so gastroparesis alone, as just that one disorder, it actually affects slightly less than 1% of the global population. That's still quite a high incidence rate. If you think of 100 people that you know, one of those will have gastroparesis. That's quite high. However, it's very closely linked with diabetes, and we know that diabetes is a global epidemic at the moment. It affects about 14% of people worldwide. It's growing very rapidly, so gastroparesis is similarly growing, and it's often actually underrepresented. It's 44% more common in those diabetic individuals, so again, similar with that epidemic, it's growing along with that. And then if we look at not just gastroparesis on its own, but gastrointestinal disorders more broadly, of which many of those are applicable to what I study, they affect up to 40% of the global population. Now we're talking 2 in 5 people. If we look around this room, how many people are in here? That means we've got a handful or a couple of handfuls of people who likely have gastrointestinal disorders or symptoms or challenges. And then looking from just an economic point of view, it's hundreds of billions of dollars, looking at stats from the US alone. So worldwide, this is a really major issue. There's major resources that are contributing to this. There are many, many people who are affected. And I think on the personal level, and that's one thing I like to connect to, is looking back to Ruby, that's what those individuals look like, and that's who those individuals are. So hopefully I've convinced you that this is something that we should care about, that I care about, that I think is important to study. So looking in gastrointestinal health, so the more specific field that I study in particular is the electrophysiology, or the electrical control of the stomach and intestines. And what we're going to see here is a view from inside the stomach. So this is from an endoscope, or a camera that's been placed down the throat inside the stomach. And we can see here ring contractions that are propagating down the stomach. The stomach contracts in a ring. That ring moves down the stomach to the distal end and terminates at that end. So these contractions are what's responsible for moving food in your stomach, for breaking down food, and for moving it between organs into your intestine. Similar contractions occur in your intestine and move those down. Very similar to the heart. I think it's always nice to draw parallels in health and in medicine. So very similar to the heart, these contractions are triggered and coordinated by electrical waves that sweep through the muscle. So with your heartbeat, the actual contraction of your heart is triggered by electricity that causes your heart muscles to contract. We see that quite often on, say, medical shows on TV. The first thing that happens, you get a couple of electrodes on and you see that beep, beep, beep. That's recording the actual electrical rhythms of your heart. And we can do similar in the stomach, but it's much less developed than in the heart. So within the stomach, there's specific cell types called the interstitial cells of Cajal that are responsible for generating and propagating these electrical rhythms that coordinate the stomach. And then those are closely coupled with the muscle cells as well. And these are some images of the interstitial cells of Cajal in red, distributed within the muscle tissue, and traces showing that the interstitial cells of Cajal are the drivers of that activity, and they then drive the smooth muscle cells, or the muscle follows that activity. So I like to just sort of dig down to that detail, and I promise that's about as detailed as I'll get in this talk, because I think that's nice to know what we're looking at. We're looking at manifestations of that cellular level within the stomach. So if we look a little bit more historically, and it was lovely seeing Amy's talk because there's many parallels, I think, and I promise we didn't talk to each other ahead of time or plan this. But I always like that quote as well, that we're standing on the shoulder of giants. I think it's lovely to look at where we've come from historically, and I think science and medical history is really interesting. So this electrical activity in the stomach is attributed to, the discovery of it is attributed to Professor Walter Alvarez. He's a fascinating man. He's a professor and also a clinician based in California in the USA. And we can see here a figure from one of his very early papers in 1922, where he's showing for the first time the electrical activity in the stomach. So there are a few things missing. We don't know the amplitude or the time duration of those signals, but what we see, those little spikes in A, are the electrical rhythms that he's recorded from the stomach. And you can see a little sketch in that lower left-hand corner of a diagram of the stomach with those lines representing the electrodes that he's placed on the stomach. He's placed some high on the stomach, and that's the top trace, where we see very little or no electrical activity, and he's placed some lower on the stomach, and that's where we see those very nice peaks that are showing the electrical rhythms that he was able to record. I love this data because I'm an engineer, I'm a bit of a nerd, I'm a bit of a tech nerd, and that is still to this day, 100 years on, one of the best electrical signals or traces that I've seen. So using what he did, it's really, really impressive. But now I get to use the magic of time travel, and we fast forward almost 100 years to the early 2000s, which is where I was doing my PhD and joined the research group that developed this technology. And we can fast forward from those couple of electrodes placed in the stomach, like Alvarez did, to now placing hundreds of electrodes on the stomach simultaneously, which allows us to look at the actual pattern of this electrical activation. Again, similar to what they're able to do in the heart, we are now moving and pushing into in the stomach at this stage. So on the left-hand side, you can see some of our electrode arrays. So there's 256 electrodes in that patch. It's 6 centimeters by 6 centimeters, so it's a relatively small patch. So it's a very tight grid of electrodes, there's 4 millimeters of spacing between those. So each of those dots is an electrode. We're able to record signals from each of those. And there's a couple of important design characteristics, and again, this was all my colleagues who came up with this. But one, those are flexible, which means they're able to adhere to the curvature of the gastrointestinal tract. And we saw in that video, it's very flexible, large contractions. So these electrodes are actually able to conform to that. And they're also sterilizable. So they're effectively plastic with electrodes, ribbons in. So the fact that these are sterilizable allows them to be translated into human patients in an operating theater, which we can see in the middle. This is in an operating theater. This is actually an Auckland City Hospital across the road. And you can see those sort of tan cables in the middle, and those are going to electrodes that have been placed via surgery onto the stomach. On the right-hand side, you can see a mathematical model or a computational model of what we saw in terms of the recordings. We're able to use these electrodes to look at the actual spatial distribution of the electrical activity in the stomach, and we can see that that actually looks quite similar to those ring contractions that we saw earlier in the video. So in the stomach, we have a single dominant pacemaker region, or where the activity originates. It's on the right-hand side, or called the greater curvature of the stomach. We get the initiation there. It rapidly forms a ring of activation that moves down the stomach and terminates at the end of the stomach. So those are all data that we were able to generate using these electrodes placed in patients. And again, this is another way to look at those data. So we can see the electrical activity moving down the stomach in those wave fronts in a healthy individual. Now, what we did from here is we also looked at diseased cases, patients who had very severe stomach disorders, like Ruby Hill with gastroparesis. And what we found was that there was highly abnormal electrical activation in those patients. So on the right-hand side, you can see a recording from one of those patients. This is a patient with gastroparesis, actually. And what we see there is we don't see those coordinated rings of activation moving down the stomach. We see quite chaotic activation. It's not originating from that single source where it normally does. It's propagating very rapidly in the opposite direction, effectively propagating up the stomach. And so we found that these abnormal rhythms, or they're called dysrhythmias, we call them, these abnormal propagation patterns were present in almost all of our gastroparetic patients, and also another disorder called chronic nausea and vomiting, which is exactly the same as gastroparesis, except the stomach empties normally. So what we found is that these electrical rhythms are quite prevalent in these patients. And that gave us, one, an opportunity to look at, can we detect these as a diagnostic to offer an early and more efficient way to diagnose these patients? And then secondly, can we correct them as a treatment option? But there were a couple of challenges with this approach. So one, it's surgically invasive, which I showed you previously. That substantially limits the applicability. Obviously, most people don't want to go get cut open to try and diagnose their stomach disorders. And then secondly, there's no current therapies for this type of activity. So that's something that we needed to develop on our own. And so just in terms of the dates, another small in the publications that we published, but that's 2012 and 2015. So it's about a decade ago, a little over a decade ago. And so those sort of challenges are what have informed the last decade of research. And so that's what I'll move into now throughout the rest of the presentation, is looking at different gastrointestinal diagnostics and therapies, specifically targeting this electrical control of the stomach. And Amy counted down in her talk. Again, many parallels, although I'm going to count up. And this isn't an actual number. So first we're going to look at minimally invasive methods for monitoring and recording this electrical activity of the stomach, so moving away from surgical invasiveness. Secondly, we'll look at gastric ablation as a possible therapy, and don't worry, I'll let you know what all of those terms mean. Thirdly, we'll look at new applications of electrical mapping of the stomach, where are we moving in the future, and what are some new avenues that we're looking at. And then lastly, looking at precision medicine to inform personalized therapy. And again, that draws nice parallels with Amy's talk around the use of new technology and new computational aspects. So first off, looking at a new potential diagnostic in terms of endoscopic mapping. So the main research question here, if we really boil it down to a question, is can we map the electrical activation of the stomach endoscopically? So that means going down the throat, minimally invasive, no cutting, into the inside of the stomach. And so we started off by designing a device. We're able to borrow a lot of this technology and techniques from our friends in the cardiac field. Like I said, there are many parallels with electrical activity in the heart. And so we're able to use a catheter that they actually use in the heart. And you can see it here. It's got 8 different splines and 8 electrodes on each of those splines. So 64 electrodes in a sphere. What we're able to do in the stomach is add a balloon. You're not able to do that in the heart. The heart doesn't really like when it's not able to fill with blood, but we're okay to do that in the stomach. So we add a balloon to the inside of that catheter. That allows us to get very good contact with the inside of the stomach. And then that whole catheter in the bottom left, you can see it actually pulls down and narrows right down. So we're able to drag that down the throat with an endoscope, place it inside the stomach, inflate it, which we can see on the right-hand side from one of our early trials in a patient at Auckland City Hospital, again, right across the road. So that was our device. We started off doing preclinical validation. Again, it's very important to validate this stuff before we go putting it in patients. And we're able to show that we can record activity from inside the stomach on the bottom side, the mucosa, that's the inside of your stomach. And that compared very highly or very closely with our normal surgical electrodes placed on the outside of the stomach or the serosa. So we're able to validate that we're able to use this device to record data, that the data is accurate based on our surgically invasive gold standard, if you will, measurement techniques. But one of the challenges was we were generating a lot of data, and the data was quite a different structure than what we were used to. So that was a little bit of a step back now to the engineering side, and we developed custom software. This is a PhD student, Alexander Chan, who, as his PhD, developed an entire custom software program and platform to be able to analyze the data from this new device, the unique spherical geometry, the different electrode orientation, and to allow us to actually analyze those data. What we could see then is that that program allows us to create these maps, again, showing the movement of this electrical activity, in this case from red to yellow, moving down the stomach, and to then push this device into larger scale trials. In this case, it was moving into a first-in-human trial in clinic. And this is a photo of us. This is the first ever patient that we put this device into, again, across the road at Auckland City Hospital. That's me on the right, a younger version of me. And then we've got a gastroenterologist on the left, some great nursing staff, and a patient lying on the table. And you can see on the monitor in the back middle of that photo our devices in the inside of that patient's stomach. So that was quite an exciting day. I'm very thankful to Greg O'Grady, who's a close colleague and friend, who was in the room and took that photo because I didn't think of it, and he had the presence of mind to think, wow, this is a really fantastic step. It's one of my favorite photos because, to me, what I've always dreamed about in biomedical engineering is developing something new and putting it into clinic, and this was the day that that actually happened. So it was really exciting to do that. We then moved that on, so we did a feasibility trial of 13 patients, like I said, at Auckland City Hospital. David Robotham is a gastroenterologist that we worked very closely with. He's absolutely fantastic to work with. And so we completed that trial and published it last year. We're able to record electrical signals, and in this case we can see where we're able to see where that activity actually originates at that star. So this is looking at the actual electricity at each of those electrodes. The red dots are where the actual event is, where the wave meets the electrode. And then we're able to create these maps. So again, this shows the electrical activation from red to yellow. And again, marked with that yellow star, that's actually a site of activation that in this case is low in the stomach, which is an abnormal activation pattern like I showed earlier with our surgical mapping. So what this shows is that even in these very early trials, we're able to actually detect these abnormalities with this minimally invasive technique, which was a really exciting outcome. So can we map the electrical activity endoscopically? Yes. So the next steps, can we correlate that activity with symptoms? And this becomes a really important question clinically because that's how we can push then towards a diagnosis. Long story short, we're not quite there, but we have secured most of the data. We're looking at finishing that and analyzing those data at the moment. In that case, we've moved or expanded to a multi-center trial at both Auckland and Christchurch, working with a second gastroenterologist, Gary Lim. Again, really fantastic, and we're working on those, finalizing those data in the manuscript now. But it looks like we can start to draw some nice correlation between symptoms and electrical activity. So that was a great outcome. We moved towards a minimally invasive option for diagnosis. So moving on to our next sort of point is, can we use this electrical activity as a therapy or as a treatment? And in particular, looking at gastric ablation as an option for that. So if we look at ablation, again, borrowing from my friends in cardiology, if anyone's here who works in the heart, thank you. So ablation is a technique that's very, very commonly used in cardiac therapy. Effectively, ablation is delivering energy, focused energy to the tissue to effectively burn the tissue or damage the tissue in a way that creates a scar that's not able to activate electrically, and that can be targeted at those abnormal sites to eliminate the abnormality and allow the normal propagation to restore. In the heart, it's done by running a catheter up typically through your femoral artery in your groin, up into your heart, targeting that to abnormal activation sites and eliminating those. We effectively borrowed all of these techniques from our friends in cardiology and moved them into the stomach, which hadn't been done before, despite many of the parallels. So again, boiling it down to a single question, if we could, can we use ablation to eliminate abnormal electrical activation in the stomach is what we were looking at. So this is another PhD student, Zara. She had a fantastic PhD, and she tackled this question or this problem for her PhD. So we did this by initially validating the set of parameters or energy doses that were effective for blocking the electrical activity in the stomach. We then set out to target sites of abnormal activation. We initially mapped the activity of the stomach using the techniques I showed earlier. We're able to identify abnormal sites in a subset of, in this case, it's a preclinical model. We use pigs in that case because they have very similar stomachs. So we're able to target sites of abnormality. We're able to mark those sites, then target them with ablation. So in this case, we performed a box ablation around that site. We then mapped after the fact, and you can see an image from us doing that. So again, this is open surgical case. And what we're able to show, I realize this is quite complicated, but effectively on the top, if we look at that nice rainbow map, the activation is in the red region. That's an abnormal activation, low on the stomach compared to where it would normally be. In the bottom, we've got that nice black box which represents our ablation, and we can see that the red activity is coming from the top to the bottom in the stomach, effectively showing that we've eliminated that pacemaker region, that abnormal region, and we've restored the normal propagation, similar to those animations that I showed you early on. We published these data, and I always like to show that journal cover on the right because that's actually, Zara's a fantastic artist. That's actually, I think, her true passion. She sort of fell into bioengineering as something to do when she wasn't doing her art. And so she actually designed, that's artwork that she designed. It's a tessellated image of stomach tissue that she then colored based on different types within the tissue. And we submitted it to the journal, and they ended up running it as a journal cover as a bit of a sort of metaphor for the connection between the gut and the brain as well, because it looks quite similar to brain images. So I always love, I talked about the personal connections and the motivation and people. I think it's really lovely seeing, especially students, bring their different passions into the work. So that's just something that's fun that we sort of did along the way. So can we use ablation to eliminate abnormal electrical activation in the stomach? Yes, we can, which again was very exciting. But one of the challenges here is those were all done in single-day studies. So if you talk to especially clinicians in the gastrointestinal tract, they get very nervous when you're putting heat and you're burning the gastrointestinal tract. That makes them very uncomfortable, which I learned very early on. And rightfully so, because there can be some really catastrophic effects. And actually, one of the major complications of cardiac ablation is if you get too deep of a burn that goes through to the esophagus, you can get issues with your esophagus afterwards. So what we wanted to then look at, and this is really, really critical if we look at the translation of this technology, is does it remain safe and effective in the post-surgical recovering period? So we set up a series of studies where we extended them out to 2 weeks post-surgery. That time frame is quite important because typically if you have a post-surgical complication in your stomach and you have a perforation, that's the most severe, where effectively you have a scar that rips open. That typically happens within 4 to 5 days post-surgery. So we're able to cover that period and then extend out to 2 weeks, which is also when surgically we see a reconnection of those electrical networks. So if we cut the stomach and rejoin them together, they reconnect after 2 weeks. So we're also interested, not only do we have safety in terms of a lack of perforation, but do we get a reconnectivity of that electrical activity, because that would not be good for us. We want to eliminate that activity long term. So we did a very similar study. We performed an ablation. We recovered for 2 weeks. And we showed that in these cases, we still had that conduction block present after the 2-week period. We didn't have any adverse complications during the post-surgical recovery. No perforation, no signs of progressing towards perforation. We were well on the way towards the healing process. So again, that was exciting because that shows that this is potentially translatable longer term. But again, and not to be a bit of a downer, but there is a challenge here in that it's invasive surgery. And so that sort of motivates our next step. But first, does it remain safe and effective? Yes. I always like to celebrate these wins. So then, as I said, it's still surgically invasive. So the next question was, can we perform this ablation endoscopically? So talking about our mapping endoscopically, can we also ablate endoscopically? That'd be pretty cool, right? So again, got a great PhD student on board, Ashton. He tackled this problem for his PhD. Recently defended his PhD, and he's graduating in a couple of months. Had a fantastic outcome, so that was really exciting as well. So looking at what we did here, we used our gold standard invasive electrodes. So we actually did perform surgery on these animals to record the electrical rhythms from that serosal surface, that surgical side. But we performed all of the ablation endoscopically. So we placed the ablation catheter down the throat into the stomach endoscopically, minimally invasively. We performed ablation, and then we also collected the tissue to look at what that ablation did and the damage done. So we can see an image on the left. That's the ablation catheter on the inside of the stomach from the endoscope, performing our ablation. That little bleached white tissue is the actual burning of the tissue effectively. And then we can see some results on the right-hand side. So these are a little bit challenging to talk through, but again, we start at the dark red. We see in the baseline that we have propagation from the top to the bottom of the stomach. Now in this case, we actually induced an ectopic pacemaker, or induced an abnormal pacemaker with pacing, so that we could ensure that that was present and ensure that it was in the same location. We then targeted that location with ablation, ablated in that location, and on the right-hand side, you can see we've got our ablations in the middle, and we've restored that activity, dark red at the top, moving down the stomach to the normal pattern. So we're really excited about this because we've now translated not only our mapping to go endoscopically, minimally invasively, but also our ablation technology. And we're carrying this work on as part of a new grant funded by the Health Research Council. So again, very excited with that work. So can we do it? Yes, we can. You might see a theme here. It's nice to celebrate the wins occasionally. So moving on, and we're getting close to wrapping up, so I think we're on time. But looking now at sort of our third aim, our third talking point for today is looking at new applications. One of those being the pylorus. So the pylorus is also called the pyloric sphincter. It's the barrier between your stomach and your intestine. Now, it's very important because that's how your stomach empties. It controls your stomach emptying. There are a whole range of disorders where the pylorus malfunctions, and we're interested in understanding, can we develop or apply some of our technology to those specific disorders? One of those cases is in post-surgical complications. So a couple of my colleagues there, I mentioned Greg, he was the one who took that lovely photo of me in the endoscopy theater, and Tim Wang, who was a PhD student as well, and did this as part of his PhD. So we had quite a unique opportunity where a patient presented to clinic who had previously had a surgery in their stomach, and I won't get bogged down in the details, but effectively they had the bottom of the stomach cut out and the top of the intestine. They were then rejoined. So what that does is it actually removes the pyloric sphincter, and you get a reconnectivity of the stomach to the intestine. The reason this is important, especially from our sort of nerdy little focus in the electrical activity, is that not only does the pylorus act as a barrier, a mechanical barrier of food and contents, but it's also an electrical barrier. So the electrical rhythms from the stomach don't propagate through the pylorus, and the electrical rhythms from the intestine don't move up. But when you remove that, we were interested in whether, potentially, some of the symptoms for this patient were caused by abnormal propagation, or whether there was abnormal propagation in the patient. We can see these are images of the reconnectivity from A being the stomach remnant, B the scar tissue, and C being the intestinal remnant. And it's quite interesting. It always looks the opposite way around because the intestine is typically quite narrow and the stomach is typically quite large, but we actually saw an enlarging of the intestine as part of the outcome of this. So that's just, again, a little nerdy aside. But then, again, we mapped the electrical activity in this patient, and we did see that red line indicates the scar tissue. This is a scar that's 20 years old, so it's a very long-standing surgery. We saw activation in this patient coming from that intestinal remnant through the scar, moving up in the opposite direction, upwards the stomach, or retrograde up the stomach, and then that carried up higher in the stomach remnant, continued in the retrograde direction. So we saw consistent abnormal activation in this patient caused by the removal of that pylorus. And we sort of framed that as a gastrointestinal aberrant pathway, or this post-surgical complications that can create abnormalities. So that was an interesting finding and discovery on its own right. And I think if we look back to the ablation technology, there's an opportunity there to actually try and reintroduce that conduction barrier at that site, potentially as something to look at in the future. But we were also quite interested in looking at the pylorus in more detail, so we developed some custom electrodes specifically informed by the anatomy of that area, because that's one of the technical challenges of recording. And we're able to record in quite high detail the electrical activation of not only the intestine and that proximal intestinal pacemaker, the electrical conduction gap at the pylorus, and then also the activation at the stomach. We also correlated that with mechanical recordings placed inside the lumen of the intestine. And again, this was a fantastic PhD produced by Sam. So I'm looking at trying to understand more about function of the GI tract using some of these techniques. Which brings me to our last talking point, being precision medicine for personalized care. So one of the challenges in medicine is that we often have a one-size-fits-all approach or a one-size-fits-many approach. But every patient is different. They all appear different. And I think particularly in the gastrointestinal field, where we have relatively few symptoms to diagnose, and we have very few biomarkers that are known, so very few things that we can actually measure from these patients in comparison to some other medical fields, we end up having relatively heterogeneous patient populations. So on the left-hand side, we can see we can separate patients out into, say, irritable bowel syndrome and functional constipation. But what we actually have in effect is we've got a whole range of patients that have different disorders or different physiology within those. So our question is, can we use technology, in this case AI, artificial intelligence, it's sort of a catch-all. We hear a lot about that these days. Some good, some not so good. But what we wanted to do was use very specific computational techniques to see if we could try and separate out with more accuracy these different patient groups. And we're able to do that using a pretty unique data set with some colleagues in the National Science Challenge on High-Value Nutrition. And again, that was work led by a fantastic collaborator that I work with, Jared Dowrick, who's leading a lot of that. And then also looking at different ways of applying some of our engineering techniques, and in this case, using computational models. So can we actually, on the left-hand side, I realize it's a very complicated figure, which Jared apologized about as well, but I think that the take-home point is that we're able to actually represent many, many different processes of the body. Each of those little squares in that figure is a different process that we're able to represent mathematically. We're able to then link those mathematical representation of the different processes to build effectively a system or a physiological system within the body. So the end goal is to create these digital twins which represent the human body and allow us to test digitally or computationally these different interventions. That's important to me. I'd like to do less work, say, with animals in the preclinical setting. Very important to do that work, but we always like to minimize that and to get the maximum efficiency from our results. So this allows us to then input some of our new discoveries into these computational methods, build these digital patients to try and improve the precision of our healthcare. And again, that's great work with Jared Dowrick. So I think just to wrap up and I guess re-summarize what we've talked about, we've looked at new diagnostic options in terms of going endoscopically down the throat to detect these abnormal electrical rhythms and these very severe gastrointestinal disorders. We've looked at developing a new therapy that can actually eliminate the abnormalities that we've seen in these patients, and we've pushed that towards a minimally invasive endoscopic approach as well. We've looked at new applications, particularly around the pylorus and the role of the pylorus, and where we can move towards that in the future. And we've looked at applying a lot of the different engineering principles into improving precision health. And I think those are each represented and were presented as independent pillars, but I think, as I touched on earlier, we can see many parallels here in terms of the endoscopic diagnosis, feeding into the endoscopic treatment, the fact that we found the pylorus could actually benefit in some of those post-surgical cases from some of the treatment we developed, and then we're able to actually amplify the impact of this using the precision health. So we can see that this is actually not independent silos of research, but a whole program that we're really excited to develop. And I think it all centers on, again, those patients. So linking back to Ruby and the team that worked with her and looking at that. Can we help Rubies? And I think the answer is yes, which is exciting for me. So thank you to many of the collaborators I work with. Some of those you'll know if you work in the university, some international collaborators, and this is really just a few of the highlights from some of those key studies that I talked about. A lot of different expertise there. So engineers, a lot of engineers on the left, some great technicians as well. So Lindley, I always like to highlight her. She's a technician that we work with. She's fantastic. We always joke that she could probably do all of our studies better than we can do them because she's watched for decades us do that work. Some great clinicians based in New Zealand and some fantastic academic clinicians overseas that we're very fortunate to work with as well. And then lastly, just a thank you as well to all of the funders. I think particularly tonight, that top line, Joe Hill from the Ruby's Voice Trust has been a supporter of our work recently, and absolutely the Auckland Medical Research Foundation tonight, and in parallel, the Kelleher Charitable Trust, who funded some of this work. And I think this is the last slide, so I think it's, again, an honor to be able to present tonight. But I dug this up, so this was an email. If you can see the date, it's Tuesday, 8th of November, 2016, at 11 a.m. I first thought it said 11 p.m., and I thought Hannah was working late that night, but 11 a.m., and it says your AMRF application has been approved. And so this application was for my first postdoctoral fellowship, or my first independent fellowship. which was just under $200,000, and that funded the very initial foundation of the endoscopic electrical mapping. So I think I'm always very thankful to the AMRF. I always remember this because this is effectively the funding that launched my career. And I hope it was an exciting journey to sort of look at what that 2 years and that funding amount ended up building into over the next decade beyond that. And like I said as well, I was very fortunate to serve on the medical committee for many years. I stepped down earlier this year, mainly due to that 2.5-year-old that I've got running around at home. But they all know that I'm only a phone call away. So yeah, thank you all for coming tonight, and I'm happy to take any questions.
Thank you, Tim. Another amazing presentation, and I think behind Tim's work, for as many yeses as you saw up there tonight, there was probably thousands of no's as well. And I think Tim's passion and his drive comes out as to what happens to make a yes occur. And behind that is a person like Ruby, both for Amy and for Tim. They are working tirelessly for people, for our health, for our better health. So we can't thank either of them any more than we can. So thank you again.
Sue.
Hi Tim, thanks, great talk. I was just wondering whether you're developing a pacemaker for the stomach.
Oh yeah, fantastic question. So just for the audience online, the question was whether we're developing a pacemaker for the stomach. The short answer is yes. I think that's another sort of parallel technology that we can borrow from our friends in the cardiac field of actually trying to drive the electrical activation of the stomach or pace the electrical activation of the stomach. So it is, we've been working for, again, about a decade on that. We've developed some custom pacemakers for the stomach, some worked on different settings that are effective for driving that activity. We have done some initial trials in human patients, and then that's been translated as well to the intestine. And I didn't really touch at all on the electrical activity of the intestine. That's actually where I did my PhD, was in the intestine. And then I moved up to greener pastures in the stomach. But looking at, there's some good work going on with a recent PhD graduate looking at intestinal pacing as well. So that's done by many of my colleagues. Leo Cheng, Professor Leo Cheng at ABI is leading that. But yeah, great question. It is an area that we're looking at.
I just wonder how many abnormalities somebody might have in their stomach. Maybe there's probably more than one, wouldn't there be?
Yeah, great question. Again, for those online, the question was around how many abnormalities someone may have in their stomach. So it's a fantastic question. And we have looked a lot at that, at actually classifying the different types of abnormalities. There are many different types of abnormalities. Historically, if we look back, it was largely looked at the frequency of activity. So how many actual electrical waves per minute are moving in the stomach. And that was, again, largely because of the technical limitations. So if you're only able to record from one or a couple of locations on the stomach, that's all you can really look at is the frequency of that activity. So again, similar to cardiology, they looked at elevated stomach rates and lower stomach rates, and there were patients that were in either of those. What we found in our work, and one of the really important factors, is that actually in many of those patients with normal frequency pattern, we found abnormalities in the actual spatial propagation. So like I showed, there are abnormalities where there's activation low in the stomach and that's propagating up the stomach. That's an example that many of those patients have the normal frequency, but it's very, very different to the normal pattern. So that was one of the benefits of this higher resolution approach. Looking at actual incidence rates, that retrograde propagation, it looks to occur in about 30 to 35% of patients. So it's quite a substantial cohort that have that particular pattern. But that's, again, an ongoing area, is looking at these individual patterns. What can we detect? What can we diagnose? And what does that mean in terms of patient numbers? Yeah, great question. Thank you.
Thanks, Tim, for your good, clear presentation. Learned a lot. Just wondering, ethnically, are there certain ethnic groups more predisposed to stomach disorders than others?
Oh, yeah, that's a great question as well. So the question was, ethnically, are there certain ethnic groups that are more predisposed to higher rates or higher incidence of stomach disorders or gastrointestinal disorders? Very good question. I don't know too much about that. I do know that there are higher incidence rates in Māori, in Pacifica. Those are groups that we look at a lot in New Zealand, and there are higher incidence rates in those groups. I'm not too sure about other ethnic groups. I do know that, interestingly, there are much higher incidence rates in females compared to males. And particularly if we look at gastroparesis, that's actually a disorder that affects females at a much, much higher rate. I think it's actually about 3 or 4 to 1 compared to males. And also affects young females as well. And I think Ruby's an example of that, of how that disorder is affecting that certain population. So I think, again, it's an area of interest looking at those different demographics. Something that we're not really looking at the incidence rates, but I did mention that we've partnered with a number of Māori colleagues, and I use colleague in a very loose term because my wife is one of those that I get to work very closely with now. But looking at not necessarily the incidence rates, but what can we learn from Māori culture and Māori knowledge in this area. And one of the interesting things we've found is that we talk about the gut-brain axis. So a lot of what we study is this connection between the gut and the brain and how those electrical signals are integrated. This gut-brain axis it's a real hot topic in the field in sort of Western medicine now. But if we look at Te Ao Māori and Te Reo Māori in particular, we can see that there's a real connection with the gut and the brain. So an example of that is the word for the stomach in Māori is puku. The word puku is also a root word or an accentuating word for many emotional terms or neurological type terms. So pukuriri means furious. So there's this sort of inherent connection in the Māori language between the gut and the brain. So we're starting to look at that and how that looks from that Te Ao Māori approach, and how we may be able to use some of that Mātauranga Māori as a treatment or as an intervention as well. And I say we as well because my wife's leading that work, so yeah.
Hi, any thoughts on the reason why the activation site is upside down or in the right? What causes that to happen?
Oh, yeah, that's a very good question. So the question was, any thoughts on why the activation site is the opposite way around in some of those cases, where the activation, that electrical activation, is actually low in the stomach rather than higher in the stomach? Long story short, no. There has been a lot of work done over many decades, and even with some of the lower resolution techniques, there were people who were hypothesizing decades ago that there was activation from low in the stomach, and there was actually, some people thought, a secondary pacemaker region lower in the stomach. What we do know is that there's no difference in the cell type, so they are consistently the same cells in those locations. There is a different inherent frequency that they operate at, so there's more proximal cells that higher up in the stomach have a higher frequency, and that causes them to drive and entrain the cells lower down in the stomach. So effectively, that's a mechanism by which that pacemaker originates high. We know that disruptions to that network can allow activity lower in the stomach, but it's really an area of ongoing research why that occurs.
I think both the presentations tonight have, they've said so much in the presentations and said it in such great terms, in terms of lay terms and us being able to understand it, that the range of questions just haven't had to be asked. But I would like to invite both Tim and Amy back up here just so we can say a big thank you. Both Tim and Amy have received fellowships from Auckland Medical Research Foundation over the years, but these two are the reason we exist. We exist to support particularly the career pathways of researchers, and that's the reason we can have so many yeses to these really, really complicated questions that are being asked, and the years and years of work and passion that goes into it is absolutely extraordinary. So I'd just like you all to put your hands together to thank these two extraordinary researchers. Thanks everybody online for joining us tonight, but thank you everybody too for coming in person to this third speaker series event, and thank you too for our 70th partners that are here tonight. And yeah, please keep on supporting Auckland Medical Research Foundation, because actually these are the people that you support it. So I wish you good night.